So as of now, we have seen data normalization, exploratory data analysis, and many of those concepts will be used like not only in the beginning, but even after differential gene expression. And that's the part now we're getting into is the gene set analysis. So now that we have a list of genes that are interesting for us, the next goal then is to find some function behind it or some relevance for those genes. And that can be done in multiple ways, which I'll go through a little bit here. This presentation was firstly made by Leif Wiege, a former NBIS employee as well. And I took the liberty to update that into this one. So what is gene set analysis? So gene set analysis is the art, let's say, to try to group genes into categories that are meaningful to us. And those categories are also defined by us in that sense. So all the databases that we'll be looking at, they are curated by people and by the science that we, and the knowledge that we gather all over those years. So the idea is to transform these gene level data, so the increase in fold changes between your groups, and transform that data into a gene set data. So for example, see that inflammatory pathways or immune response is downregulated or upregulated for, in this case of pyruvates and so on. The gene set per se can be many things, and they can be many mean different things depending on what actually you're looking at. So for example, like gene ontology terms, pathways can be cell types, chromosome location, transcription factors, histone modifications, diseases, and so on and so forth. But here we'll focus mostly on the transcriptomics and differential gene expression analysis that we have done in the course. But actually you can apply this to any kind of genomic data, DNA methylation, acetylation, attack-seq, chromatin accessibility, and so on. So why do we need to do gene set analysis? Because that provides us a way to interpret the genome-wide results or the transcriptome-wide results. So the gene sets are usually also fewer than the genes, the number of genes. So instead of 20,000 genes, we have 100 annotated terms. And that's much easier because all these terms, they also have more descriptive names, such as inflammatory response, processing of a metabolism or of a metabolite, and so forth. So it's of course difficult to manage a long list of significant genes. So then we can compile that into a couple of enriched terms. And also that allows us to detect patterns that would also be difficult to, if you just look at the list of differential genes, because many of those genes have exactly the same expression trend because they belong to the same pathway. And in that way, we can also remove redundancy in the data in another way, other than PCA, for example, that we have seen before. And the top genes might not always be the interesting ones because some of those are correlated or coordinated together. So some small changes also might impact into the enrichment for some terms, and those might not always be the top ones. And that also allows you to integrate some external analysis into your data, external information, I mean. And in that case is, for example, annotation or what those genes do, or how they are regulated, or yeah, it could be how they are related to diseases and many other things like this. And in this case, they are also less prone to false positives, because if you have all the signaling pathways that is upregulated, then that actually means that is a really significant result compared to just a couple of genes being upregulated and showing up in your top differential gene list. Because here now we are considering so many genes at once that the chance of false positives is a little bit lower. So what about the gene sets? There are many that you can use, and that also really depends on the research question. So there are many databases that you can use for looking for those gene sets. The molecular signature database from Harvard, for example, Enricher and Panther are very common ones. So these platforms, you can even go on them online. But most of the terms that you can find, you can download from their data set, from their website. And that also is already included in some analysis tool that you can really download. For example, in Enricher, it already downloads directly from Enricher. So the package is called Enricher itself. Or downloads directly from Gene Ontology or from KEGG, or using Biomart, as you saw also in the previous lectures. And some Go terms are usually more widely used as gene sets. So the gene ontology that I'll mention in a minute, which comes to that one. So gene ontology is a hierarchical organization of the gene terms. It's divided mainly in three categories of biological process, BP, MF, which is molecular function, and CC, which is cellular compartment. So for biological process, it means group all genes that have a similar function. It doesn't mean that they interact with each other at all, but are genes that are related to that function. So for example, neutrophil chemotaxis. You have several proteins that interact both in the neutrophil membrane and the cell matrix, or that some of those proteins are not even connected to each other depending on the tissue that you have. But all of them are demonstrated to impact on the neutrophil attraction to the tissue. And in that case, they will be grouped together as neutrophil chemotaxis related proteins. The same thing goes for cell proliferation. Some proteins are related to the same, but there are so many genes that they can be compacted together into this definition of cell proliferation. Molecular function relates to the really molecular part of that protein. So acetylation, phosphorylation, and such all functions like this. So adhesion and so on. Yeah, it can be many in different ones. And cellular compartment relates more to where that protein is actually present in a cell, or could be present. Some proteins are present in multiple locations, but here, for example, nucleus, cytoplasm, and plasma membrane. Mostly, and the more common, especially for immunologists in that sense, that will be the biological process. But if you have, for instance, cell biology analysis, then you would like to focus maybe on molecular function or cell compartment. So the terms get more and more detailed, moving down the hierarchy. So this is what a tree looks like. And then in the top, you'll see biological process. And then they will start subdividing in small and small chunks. And being small, each term will be containing less and less genes and becoming more specific towards the leaves here. And here, for example, in this case, the color indicates how significant and rich those terms are. And usually you see that the leaves here are becoming significant in this part. And some of those terms, for example, this will be, for example, family of ribosomal RNA translation. Here probably is a metabolism of a small molecule. Here, chemokines. So all those concepts are hierarchically and ordered together into this tree. And of course, just remember that some genes can belong to multiple gene ontology terms as well. So they are not specific. So you, a certain cytokine can do not only neutrophil attraction, but also attract other cells. So those terms also contain that protein. Another very common database to look into biology and function is the KEGG, which is the Kyoto Encyclopedia of Genomes. And in that one, they have different information regarding the pathways, which is the most commonly used. And in that case, the pathways actually are connected by a reaction. So not only like gene ontology that they are just cluttered together to represent that information, but now I know that to glucose to be degraded to fructose or whatever reaction in that case, there is an enzyme, and that enzyme is a protein. So I know in that pathway that all those proteins are actually connected in this graph. Like, for example, a really metabolic map, like this one in different colors representing different metabolic functions. So I know that there is a reaction going on in that case. So it's a different kind of perception how you interpret a term or a pathway. So a pathway means that those proteins are connected somehow by a reaction or some metabolite or anything similar to that. Wiki Pathways is an open version of KEGG plus gene ontology, is also related to pathways in that term, that they are connected somehow. But it's similar to a Wikipedia page, for example, that they will be maintained by the community and it's more easily accessible. But however, they can also be prone to errors as well, just as Wikipedia. But the idea is just to have a more comprehensive as well. And as far as I know, is also a good database to look into. Sometimes many curations are better in here as well. Another one is called Reactome, which is also very similar to what KEGG is, that they compile the information about many processes and a tree hierarchy as well as gene ontology. But in here we have also a more visual interpretation of the whole data set. You can use their annotation. So it's another database that uses their own annotation. And many of those annotations actually will be very similar to KEGG, to gene ontology, to wiki pathways. So just remember that if you run gene set enrichment analysis, it's very okay to run multiple of those and just take what is the consensus, what I see in every single database that I look at. You can also do transcription factor enrichment analysis or use that as a pathway. So I know that this transcription factor regulates all those proteins. In that case, that is also an enriched term that it can be taken. Instead of having inflammatory response, I will see GATA3 is enriched in my data set because many of those genes are upregulated in my data set. And another one is from the molecular signature database, which is called the Hallmark gene sets. So this is taking a public data set and analyzing them to form co-expression patterns. And those patterns are curated by the Harvard, so the molecular signature database, and compiled into these Hallmark gene sets. So it's kind of a big summary of a gene ontology, let's say. So, and those pathways, most of them will be also independent of each other, because that is a better set to avoid redundancy that is required, and that would provide better results for gene set enrichment analysis that I'll talk in a minute. So whatever database that you use, so I can refer, for example, the mouse signature database or enricher that I mentioned before. This is where you can find like Hallmark gene sets, other potential one, curated gene sets, regulatory targets, and yeah, ontology, gene ontology will be somewhere here, and many other immunological signatures that are based on public data sets, as well as for enricher, there are also many other things. So chromatin accessibility, cell lines, the Allen Brain Atlas, there are many other databases that you can look at. So here I'm just referring on the top of the iceberg, gene ontology, KEGG, Hallmark pathways, Reactome. So those are what usually come into my mind when I think about databases, but there are many, many more that you can try in your data set. So if you're looking at transcription factors, there are different ways to interpret that as well. But that's really depending on your specific project, for example. Now, how do we do the analysis itself? How do I connect those two informations together? There are mainly two methods that I'll mention for now. One is to do overrepresentation analysis, which is done using a hypergeometric test or Fisher exact test. And the idea or concept in this is to find, for example, in a list of differential gene expressed, let's say up or down, or in this case, we'll focus more on for example, only up and then do the down separately. Or for example, the list of genes in a cluster or in a module. So I just have the gene names, for example, in this case. So, and these are the genes that were selected, differential gene expressed or not, or belonging to a cluster, and the ones that were not selected. And then I check if those genes, how many of those genes are in a particular gene ontology term or not in that term. So you can see, for example, like among the 10, the selected here, 8 out of 92 are inside the gene ontology term, and most of them are inside my selected list. And that will give a positive answer to my question that these terms are actually enriched, because most of my terms are actually belonging to a gene ontology term. And here you have like the rest of the other genes. But how to visualize this a little bit better, it's simply like this. So for example, here you have the list of all the genes that I have. 8 will be around here, are among my selected list of genes. The same thing for like another term, they will have a bit proportion in here, but most of them will be not. So for example, these terms here are ordered by how much enriched they are. Since here, most of the terms are belonging to my selected list. In this one, only part of it. This one is also the same part of it, but there are many others that do not. So it had a little bit less enriched. This one, the proportion of them is also lower, and then lower, and then this one is even lower because there are many more genes. So higher chance of those genes being selected here, but they are not selected. So this is also a lower enrichment. And this is basically what you will see in your analysis. So it's a log 10 of the p-value or the false discovery rate, or some enrichment score that every tool has a different one. And then you can set a cutoff here, my p-value below 0.05 or so on. Do you have any questions at this point or about these enrichment? No? All right. So if you remember this paper.
Sorry, Paulo, may I have a question?
Yes.
What is the minimal number of genes which is reasonable to put in this kind of analysis?
What I usually filter, I filter terms that have less than 3 or 5 genes inside. So it's not the number of selected list, but the number here between the term and the list should be at least 5. But this is a very personal cutoff as well. So yeah.
Okay, thank you.
Yeah, I also have a question. Why do you say that we can either use all up or all down regulated genes and not both in the same lists? Because when we say we want to check all differentially expressed genes, why can't we do this?
Yeah, because then you're mixing the trend of the gene. So if you want to know which pathways are up, so then you select the ones that are up and do one enrichment, and then the down you do it separately. If you do all of them together, you will see terms that are enriched, but actually biologically that is not meaningful because part of your terms are coming from up and part of your terms are coming from down. So it doesn't make sense that they belong in the same pathway. So that pathway should not be enriched in that sense.
But don't pathways also include like inhibitors and stuff?
So yes, it actually depends on the databases how you can select these genes. But in general, yeah. So you have to think about the database you're using, and then when you're comparing.
Yeah.
But if it is like you say, that if you have like a set of genes where some of them are being upregulated because of something, and some of them are being downregulated, then you should use up and down at the same time.
Yeah, here in the presentation, I did not include another method that actually can use the fold changes to consider the enrichment. So there is one package, and there's also in the exercises at least mentioned the reference, which is called Piano, and that will take both the fold changes and the gene list. So for example, like if it's belonging to one gene ontology or another, and take that into consideration when doing the enrichment, and that those pathways will then take both up and down genes. But then you need some kind of information about these how much they are enriched up or down, because that will do impact on your results. Yes. And as you're probably already familiar with this picture, that is exactly the same analysis that has been done here. So you have your samples and you divide them in gene modules using hierarchical clustering, which we did yesterday. And then you take those list of genes and do an enrichment analysis. So then for each cluster, you have a different enrichment analysis done. And then you can just highlight here the top 3 signaling pathways that came out from that analysis, just to have a easy representation of what each of those genes could actually mean. And you see that actually many of those pathways are related to each other. So for example, as we saw that, for example, here in the beginning, there's response to steroid hormone, lipid metabolic process, triglyceride catabolic processes. So all of those are related to lipids, and all the lipids that are related are actually a subgroup of them. So you see that there is a lot of redundancy in the data set, in the databases where you get that information from. So the other method to use for doing set enrichment analysis, it's called the gene set enrichment analysis. So far we have been doing gene set analysis, and this one is what is called enrichment analysis. So here is only for 2 sample comparison. In most cases, you have like in our data set, we'll have a case and a control. And what you do first is to sort the genes based on their expression mean. So for example, here group A, they're up, and then in group B, they are down here. And then you have getting to the middle, they are kind of the same level. And then up to the late part, you have up in group B, but down regulated in group A. So in here, we don't have these up or down differentially expressed genes, because we don't need to set a cutoff. We just need to sort all the genes that we have in our data set based on that. And all those genes then will be parsed together to the gene set enrichment. So now consider that we have a gene set S, let's say immune response. And then we try to highlight where this gene is in this ordered list here. So we can see that many of those genes are belonging like in the beginning part. If you compare to the end, there are not many lines in here. So each line here will be a gene belonging to this gene set. So since all of those are very conserved in here, and this is exactly the same as this one, but just rotated, you perform a random walk that you start scoring the genes if they are belonging to this class, or this class means belonging to this gene ontology term. As you move forward, that increases the amount of enrichment terms that you have, and then the rest of the path will eventually go down to 0. But since you don't have that many, they will go down slowly. And in that case, this is how we consider an enriched term, because now we don't need to filter by p-value, by fold change or anything. We just have to sort the genes based on their abundance when you compare those 2 groups. But how does it look like then? So for example, here we have a term that is defined in here, and then this is in the enrichment curve that we see. And the enrichment itself is just the height, the maximum height that we have in here. But this is just a small detail. What we need to look at is the curve. So the curve is like shifted to one side, just as this one as well. So these terms are enriched. In this one, for example, it's not enriched because now we have, rather than a complete shift, you have like a normal distribution in that sense. So it's enriched in the beginning and then enriched in the end. And you can see that the genes here are evenly distributed across all the ranked gene lists that I used as input. While these ones, the genes are more highlighted in the beginning. Likewise, if you select a gene set that is downregulated in the first comparison, because here is always comparing A versus B. So now if it's B has more than A, then I can use an annotate here, draw. So then you'll have exactly the opposite, which will be a curve like this. So this will be a downregulated gene pathway. This is up in the first group, and this is down in the first group, or up in the second group. And I can't write down, which is probably wrong. All right. Any questions on this one? You will have more exercises on that as well, and some other examples. So one thing to consider is that there is a lot of genes that will be overlapping in all those pathways that you will see. So many of those terms will also be present there. So because of these high overlap of genes belonging to many pathways, it is possible that when you do a differential gene expression, you have many significant genes, that the top pathways will be relevant to one pathway. Maybe like 10 terms will belong to the kind of same pathway, let's say lipid metabolism. But actually, there are still other terms below in that list that might be related to something else, but their p-values will not be that significant. But it's still, that could be still a biological result for you. So always take care to also look down in the list and check for other patterns that you see. Maybe you see antigen presentation, regulation of MHC, or something, that all pathways that are very similar as well. So that might be hindered because of these like highly enriched pathways. It doesn't mean that the ones that are below do not occur. They are just less preferentially taken into the enrichment scores. So always check for that. So then you're not missing any important biological information as well. But of course, if they are in the top, there is a reason for that, because their p-values and the distribution or their enrichment scores are actually more significant than the others.
Can I ask a question to this?
Yes.
When you say that we should also check the other results lower on this result list, but this requires that we have a certain hypothesis about what we are looking for, because otherwise we could look for everything and then everything will be interesting because we have no idea what we should look for.
Yes, so in many cases, RNA-seq is more exploratory than answering a question. So you start with the RNA-seq, you have new findings, and then you validate. Because if you do, for example, I have done experiments that we treat mouse with inhibitory drug for cholesterol biosynthesis, and we see that is actually the difference that we see, and that's the only difference we saw. So in that case, we are satisfied with the result because that is what the result should look like. But if you look down the list, then you see other inflammatory pathways that were also showing up. So that really depends on your question in that sense. Yeah. Yeah.
Okay. Thanks.
So some considerations. So there are, of course, biases in gene set collections, because those are related to what we know. Immunology, for example, is a very well curated domain, or some parts of cell biology. But for example, host-parasite interactions are not that much. So those terms, even though they are there are probably not that many terms to show up in your differential gene expression list. That's one reason why you should look down sometimes in the differential in the list of ranked terms, because some of those might actually be meaningful to you. And always take a look with a grain of salt. So some of those genes can also be misleading because they are just everywhere. Some of those genes are belonging to many different pathways. So you need, in that case, you need to know the biology of the genes. Also consider, for example, these gene set size. Sometimes inflammatory response to immunology doesn't mean anything. Just because I'm looking at an immunological disease, so inflammatory response should be there at some point. Consider also the positive and negative association between the genes. So up regulated genes should be used together. Down regulated genes should be used together as well. and do not mix them in that way unless you have a method that can take that into account. Yeah, and remember always to revisit the gene level data as well, because sometimes some of those genes might be significant, especially for gene set enrichment analysis, that we don't have a specific cutoff. So we might find some results that are actually not that meaningful or that significant. So in some cases, always check that you have a gene that you can validate, or do a qPCR, for example. Yes.